Thank you. So I'll start by thanking the organizers and Dimple particularly to letting me in this meeting. So I'm going to discuss a little bit about what we have been trying to do to understand function in the genome beyond the genes, and we are doing this for some time from the beginning of the century. So I'm going to summarize you few things that we have been doing and the approach that we have been taking. So as we know that the blueprint of life is in the genome, but how do we understand it? How information DNA sequence leads to a biological output, and how this information is encoded, regulated, and expressed. One way of doing this is to compare the genomes of various organisms. And if you know what is different in two organisms, they are closely related, then you can go to the genome and ask what has been added or deleted and try to bring the information out of that. So that's one way of trying to understand how information is coded in the genome. So this is very common slide you may have seen. So if you look at the genomes from simple to complex, the genome size increases, so which is expected because more information is required to make more complex machines. But what is unusual is that the part that codes for the protein is becoming less and less. So per gene you require 5 kb in simple organisms, 120 for humans, because we have many cell types. So complexity is how many cell types you have. So that means same genome is now functioning in many different, giving many different functional outputs. So what you are seeing is that the outcome of evolutionary process is that the number of genes are more or less static, around 20,000 plus minus something. But the non-coding part of the genome, so-called non-coding part of the genome, is consistently increasing along with the increasing the complexity or epigenome potential of the genome. So, what could be the function in this the part of the genome which is not coding for protein? Right? So this is in a way providing grammar to use protein coding regions in many different ways, so you can make more complex organisms. So one part could be that there is a packaging code in the genome. So you have cell type specific packaging. So certain genes are not accessible and some are accessible. And that makes much simpler way of not using certain genes and making others available. So each cell type should have its own packaging. And now technology is emerging that you can actually figure out this to large extent in different ways, where there is accessibility, different kind of epigenetic modifications, and lots of progress has happened technically and even understanding the packaging. The other regulatory function that is required is that many times the things that work together, they are not insist very close by. So you have to bring things from very far from the genome, or maybe sometimes from different chromosomes. So in the packaging, these regulatory and their targets come together. So that's another way of what we can call as a long-range interactions or subnuclear compartmentalization. So relevant things come together. We know it happens. There are many examples. We don't know how it happens, but it's something very exciting. Then large part of the genome is transcribed, although it doesn't code for any proteins. There must be some other function, and many interesting aspects are emerging how the non-coding RNA plays a regulatory role in many different ways, including the structural roles. So in this background, what we decided to use as a model system to do, because we do experiments to understand these things, and the model we chose is a homeotic genes, which exist as a cluster. As you can see, it's an ancestral cluster. So in fly, you have 8 genes. Same kinds of genes are there in vertebrates also. They are also clustered in same order. And what is interesting to see is that there is a color code here that genes at this end of the complex make the posterior end of the fly, and this end of the complex makes the anterior end, and the same thing happens in the vertebrates. So nature knows only one way of deciding anterior posterior body axis, and this is through these genes, a cluster of genes. Vertebrates have 4 clusters, so invertebrates have only one cluster. What in the fly, so how this cluster is split into 2. One is antennapedia complex, I'm not going to talk about. What we use is the bithorax complex, where there are 3 genes. And these 3 genes, lots of genetics has been done, and based largely from the genetic data, we know that, so these are the genes and these are the regulatory domains of these genes, and they are separated by what we call as boundaries or insulators. And each of these cis-regulatory module contains memory elements or polycomb response elements or trithorax response elements, repressors, tissue-specific enhancers, segment-specific enhancer, promoter targeting sequences, and so on. Lots of these things, just a cartoon to show that they are quite rich regulatory domains, and they regulate the homeotic genes in a way which is shown in this fly. So when the fly lays the egg in the beginning, there are information that is also deposited by the mother. These are called segmentation genes. Most of them are deposited in the egg, and then some are early expressed, like, and then they are used along the body axis by in the egg. So these segmentation genes come and bind in many different permutation combinations across the complex, and then they, around the same time, they determine, like, this part will drive the expression of UBX, this part will drive expression of ABDB and ABDA, and so on. So what you see here is that this domain is, let's say, IV5 is driving the expression of ABDB in a level that it will make this segment of the embryo. And then when it transforms, this also same information carried in the adult also. So this regulation at this level is very nicely understood. But you also see why I am showing this to you is that other than regulatory element, their arrangement and order in which they are occurring is very nicely reflected in the phenotype that you see, or their function across the body axis that is. So it makes very interesting system which has full access to genetics, of course, all the molecular biology techniques and so on, to study organization of the genome and its function in a larger context. So this is what the kinds of mutations and then later some molecular aspects we have been using. So now this is also a way of looking at the fly. So this fly has been cut open adult from the back, and you spread and you see the cuticle preparation. So this is the dorsal end and this is ventral, and this central nervous system of the fly. So this is normal case when I mean, we have ways of identifying which one is the 6th segment, 5th segment, 4th segment of the body. And this is the staining for the ABDB. You can see that more ABDB means more posterior, less means more anterior. Now, if you delete this part, let's say IB6, then the 6th segment will get affected. And what you see the phenotype is now instead of 4, 5, 6, you have 5, 5. Right? There is no 6th segment. We know it because you don't see bristles here, you see bristles here. So there are subtle ways of knowing that there is a homeotic transformation. Also, you can see ABDB level in 11th has gone down compared to here. So there is a more interiorization of the, so 6th has become 5th segment. Now, if you delete the boundary, then the function is just other way around. Now you will see posteriorization. So we are here, if you take this boundary out, then IB6 and IB7 are now one domain, and they both make 6 and 6. 6 gets transformed into 7. So here is a male cuticle, male abdomen. You are seeing the 5th and 6th. Here you see only one pigmented segment, and 6 has become 7th. 7th is rudimentary, so you don't see it here. Right? So this is called posterior prevalence. When two things get mixed, posterior one dominates because the level of ABDB will be high, and then you will see more posteriorization happening. What we also did is we combined some of these deletions. So just to save time, I'll make it simpler. So here there's an element called MCP. If you delete, then IV4 and 5 are mixed. Now you see 2, 3, 4 is gone to 5 because you see posteriorization. So 5, 5, 6. And I just showed you the phenotype of F7, where it is 2, 3, 4, 5, and 7, 7. Now, what happens when you recombine the two things? These kinds of the game you can play in the fly. So you recombine is challenging because they are very close by. So you have to work hard, but you managed it. So when you do this, then you see the transformation is 2, 3, 5, 5. So they are interacting, but we see additional phenotype. You see incomplete cuticle formation. That's a different thing, non-homeotic function of ABDA. It led to different thing, but what it shows is that when you combine, they can interact with one another. You don't have to go in many details. What I'm trying to impress on you is that there is a system in which you can delete various combinations and then see what happens in the phenotype and then try to interpret how these elements are now interacting, because everything is because of the level of ABDB in this region. That's what is deciding how these things are changing from one to other. If this happens, this was relatively easy. What we decided to do is, so relative positioning of the cis-regulatory module is important for the segment identity and the level of Hox genes. What will happen if we deletion some of the USHA, duplicate and invert CRMs? If we duplicate, we'll see longer fly. If we invert, we'll see confusion in the fly. That was, and it took us a long time. We started doing it some early on, but later when the new technology of genome editing became available, we were able to do it very easily. So now this was possible to do, and we actually made those flies. So this is the thing I'm talking about. So deletion is here, so IV5 is gone, so you see a shorter complex. This is duplication, so IV5, IV6 is deleted, duplicated. So, and then here's this whole part is inverted, right? So this is the, and we were very excited to see what happens to these flies. But when we made these flies, so, okay, I will show you the result. So deletion as was expected, when you delete IV5, the IV5 is gone, so you will have A5, A5, and this is a female cuticle, so it's expected phenotype. Disappointment was when you duplicate, we thought there may be a longer fly, but fly is actually normal. There is not much happening to the fly. So A5, A6, A5, A6, you see, I mean, you don't have to worry about these are some small details, but outcome is that fly looks more or less normal and it is fertile and walking around. Even surprising was when you do inversion, actually fly is still normal. You see some homeotic transformations, like here A4 becomes A5, A6, 7, female also similar phenotypes, but these flies are fertile and viable. This was a disappointment for us because we were expecting fantastic phenotypes, but phenotypes were really very subtle that I have to convince you there is something different in these flies. So what we learned from this is that deletion of regulatory module phenotype is of source known homeostasis. Duplication CRM in a modular way renders variable phenotype, and relative position of CRM is largely dispensable for enhancer specificity. And why is then this collinearity so much conserved all the way from vertebrate to insect to vertebrate, although you reshuffle and actually not much happens. So that is something which is still puzzles to us. So what we learned is that number of segments don't change based on security modules. If they are deleted or duplicated, but identity changes, and CRMs regulate identity and not the number of segments. That's what I'm saying. So now our hypothesis is that number of segments are determined by the maternal effect or segmentation genes, and we can figure out some of those by, this is the kind of thing that we are doing in the lab now. So you tag ABDA and ABDB and follow them in vivo in the context of these mutations that we have generated. So this is just to show you that we have now tagged flies and we can do in vivo imaging and watch them. They are differently in situ tagged. We are also now doing the 3D organization analysis to ask which part is now interacting with the promoter of ABDB or something different. We want to recombine them in many different permutation combinations. So now we have lots of those flies, and then see if by difference in the phenotype we can figure out later what kind of long-range interaction changes are taking place. And of course, the polycomb and tithorax are the genes that are not required for setting up the expression pattern, but the maintenance of expression pattern. And we are doing extensive analysis to bring these mutants in the background of our rearranged complex and ask how much is the initiation and how much is maintenance, what we are seeing. And then, of course, I showed you the phenotype of the cuticle. Internally also there is a phenotype that is possible, very challenging to see, but we have some ways of looking at that. And this is just to bring to your attention that in Drosophila, in diploid cells, genomes are always paired. So when genomes are paired and when you have deletion and duplication, you see weird kinds of looping that happens. So our 3D experiments we are designing in a way, Suhail is here, he is putting most of the effort in this context to ask if this interaction in homozygous or heterozygous or transit conditions will give us some idea about what are the things that are happening. So this part of the complex, it shows that we have a system in which you can reshuffle, look at the phenotype and go to molecular and ask who is touching who, and then and how this result is leading to the altered expression of the target gene, which is the homeotic gene here. Advantage is they are very sensitive for the phenotype, so we know the function and we can now link the molecular aspects to this. So very quickly, I will just show you that this is not what I am saying is not really unique for the homeotic genes. The genetic and biochemical studies that are done in this, they have a consequence across the genome. And this is what we have been actually realizing and doing at the whole genome scale. We learned from about the boundaries and PREs from the Hawkes complex, but that biochemical information we use to develop tools to predict across the genome such elements, right? So we call it, if you want to predict boundaries or insulators or barriers, different people have different names. We think they are similar kinds of things. So we know what proteins interact with them, what are their recognition sequences, and then we essentially take the cluster of motifs that are DNA binding protein motifs, and then look the permutation combination across the genome and ask if we can predict boundaries and PREs. So Srinivasan single-handedly did all this. So he developed this tool. You can actually, we know enough biochemistry that we can make a reasonable prediction. So he did thousands of boundaries. He predicted across, there are 12 genomes of different Drosophila species available. We find similar kind of things. And then many other features. What is interesting is, so this is the map, and these are the boundaries that Srinivasan mapped. And then you can see that they are essentially demarcating the genes and the various epigenetic marks or the binding of these are polycomb, posterior sex common, enhancer of just binding. You can see they map this chromatin. Differently marked chromatins are separated by, and the genes are separated by such elements. So this was very interesting. So we compared with other insects. Do we see the same kind? So we did, of course, experiment and showed that about 80% of these predicted ones we could assay for their boundary function in the S2 cell assays. And even in, let's say, in this is malaria mosquito, we annotated the complex and predicted the chromatin domain boundary using the tool, and then tested in the fly, and they actually function very nicely as would be expected. So that is one way, so you can predict that is about 3% of the genome it accounts for. So Srinivasan used a similar thing for PRE. There also we knew enough about the biochemical biochemistry of these proteins, DNA binding proteins. So you can see these are various PREs, the known experimentally, and what are the motifs of various DNA binding proteins that make this PRE. So using this, when Srinivas made this PRE mapper, you can see that all Drosophila species have a very similar number of predicted PREs, about 20,000 of them. And in other insects also, when we do, we get similar kind of prediction, and except a few, maybe some proteins, they have acquired new proteins, so that's why missing some, or maybe they have lesser number of PREs, unlikely though. So using these tools, we are able to predict PRE, but are these predictions working? So here is like similarly you saw, so this is the predicted PRE, and you can see if you map with the polycomb binding or PSC binding or enhancer of just binding, you can see that the peak comes where the PRE is predicted. So that will be as you would expect. But you can also notice that there is a modification up to here, then there is a sharp decrease. So one would predict there should be a boundary here. So in fact, wherever this was possible, we have done combined the two tools, and we see that like here you have a boundary, you have PRE. So PRE has a peak of this thing, and the boundary will demarcate that this will not spill to this side. So this, and we have seen this boundary PRE combination in bithorax is very common. That's how we had some idea that it may be there. But now we see in the genome, oftentimes we see boundary and PRE combination. So that means there's a memory element. Whatever happens, it will remember for the rest of the life. But this functionality will not be leaking to the next side because there's a insulator sitting next to it. We also looked at whether PREs have anything more transcription start sites or this is other boundaries or TAT borders. And we see that there is a proximity of predicted PREs. Of course, one has to do experiments and see if there is a meaning in that. So what this shows is that we have enough of information, particularly epigenetic modification data, that you can predict the PREs and then compare with the rest of the things, other features. And you can see that most interesting is that this is a DNase1 hypersensitive site, and see the major PREs actually map to hypersensitive sites. So we feel that this gives something useful when you use the available information and make simple tools. And so here, for example, if you add up the two, then we kind of annotate 10% of the fly genome, which is quite a bit. What happens in the vertebrates? So vertebrate biochemistry is not that well understood. So we are trying different approaches. For example, for protein, for the boundaries, we are trying to find pairs of genes which are very close by, less than 5-10 kb. And actually, most of the genes are very close by. That is one thing we have discovered. There is a gene dense regions, then there are deserted regions. And we are taking these closely spaced, differentially expressed genes and searching in these thousands of intergenic regions for patterns, where there are motifs from going to epigenome data and in context and looking for pattern. And I think we are close to now to a tool that can actually predict boundaries even in vertebrate systems. So that's the work that is happening. So, and we will test some of them, and some other occasion we will talk about it. I will very quickly tell this something which we are doing for quite some time. So there was one way, experimental approach, some rationale to use, look for non-coding part of genome and try to find function in them. But then there are other parts of genome, for example, simple sequence repeats, they are there in plenty. And do they also have some function? They constitute 3% of the human genome. And Akshay and many others in the lab have worked for many years. And I'm just going to give you just very briefly how we approach this, and we found something interesting about simple sequence repeats. So these are just the plot of, so I have 5 minutes. Oh, okay. So, okay, then I have to really cut this short. So different repeats occur differently. Some are not present, some are highly present. And if you look for other SSRs, you see same kind of thing. If you look for the length of these repeats, generally you expect longer repeats should be less, but that is not the case. There is a preference for the repeats. So, so there is a certain kinds of repeat, a certain size of repeats which are more present than others. So we, with this and few other information, we thought they might be functioning something like boundaries are defining domains. And we did an experiment. So we took this repeat, put in the fly system. So this is a transgenic system. So mini-wide will give eye color. And this is Fujita-Raju enhancer will drive expression of 7 stripes. When you take this human sequence, this was the human repeat taken from Y chromosome and put here flanked with the 40 sites, you see that nothing happens to this. There is no repression, but this enhancer is not able to reach here. You can flip it out and you get it back. So, in fact, there's even more interesting. By accident, we found insertion in the arm locus, and when we flip out, we see that these enhancers, which are not able to act in the various imaginal disk, they are able to interact. So, which means that there is a simple sequence repeat functions as a boundary, and we have now tested large number of them, and a lot of them actually function as boundary in a cell system, and some of them works as repressors and so on. But so I think I have to skip this. We have done across the genome and see this means Lack of repeat, this means more repeats, and there are some places where you see uniquely abundance of repeat in this certain vertebrates and so on. So I will skip because I want to show you something else. So some repeats are species specific. So for example, this repeat got introduced at this point in the evolution. All of them have. These guys don't have. That is reflected here. So these are here and they are here. So they are now even species specific or class specific repeats. Some of them are listed here that how many millions of years they separated, and they are almost uniquely abundant there. Other organisms don't have them, right? And they are maintaining it for quite some time. So I will just take 2 minutes to tell that how this repeat then function. So Maninder is here, his poster is there, so I can go even faster in that. So Maninder, so there must be some protein which bind. DNA by itself doesn't do anything. So Maninder has done lots of gel shifts and they come up with the repeats which show a specific binding. So it will compete by itself and something else will not. Then he, by doing lots of permutation, combination of gel shift and competitions, he identified certain 8-10 repeats for which he made the affinity column, purified the proteins, and okay. And then he found several proteins. He has done, I don't know how many, some 100 odd proteomic experiments. And we found some known things like D1 is the ATU containing protein which is specifically interacts with this. Then there are Jigar1, DIP3, and so on. So, of course, they are there. There are many ways of figuring out. So, he developed, this is the last bit I'm going to tell. So, he developed an assay how you can use genetics to apply for identifying the function of these proteins that we are talking about. So, this is the locus, vestigial locus, where there is an enhancer which drives the expression, and that's why you have wings in the flies. If you take a deletion of this DV boundary enhancer, then the fly will not have the wing because there is no enhancer there. So he used this locus and added ADP containing GFP, so flies now have a green eye. But the advantage here is that you can bring in a cassette now and exchange this by any other. So here is gypsy boundary that we are replacing, but you can replace anything that you want. So when you okay, so if you bring gypsy, you have not deleted now enhancer, but enhancer cannot reach here, so there is no wing. Gypsy is known to bind to suppressor of hairy wing, so we bring a suppressor of hairy wing mutation, homozygous, phi viable, and wings are normal. So the same thing, but now suppressor of hairy wing is missing. So this opens you now you can do the genetics of the by visible phenotype. So you have a boundary. Now you can go and cross to any mutation, or you can do mutagenesis and ask when the wing becomes normal. So that means the boundary function is lost. That means you are looking at the protein that works on that boundary. That was not there before. So of course he did his experiment to introduce these repeats at the locus, and we didn't see absence of the wings, but we see notch of various kinds. And one reason of mild phenotype may be because these proteins that bind to them are not present there in large quantity. So he used again the good tools of the fly and expressed in this case, let's say D1 in the same tissues, and now we see the more severe phenotypes. So which means that if you provide the protein, you have the repeat, you can see the phenotype. You take away the protein, then you see normal wings being formed. So it opens you the genetics of fly to do this kinds of now function of non-coding part of genome. Here we are talking about boundaries, and then you are talking about in vivo role of this repeat. So I just want to say the last sentence is that now we have enough of technology, methods, data available to explore the information content of the genome, and maybe now we should be able to use with the experts the AI and ML tools to apply in this context. It will be a rewarding exercise because we know so much about the genome, but we cannot figure out how this all this regulation happens. But I think we are reaching at this point with all this information. With that, I'd like to thank you all again and thank TICS and CCMB. The work I did was all done in is being happening in CCMB, and DST for JCBO's fellowship that supports my research in Hyderabad. So thank you very much, and happy to answer if there are questions. Maybe I should have less information. I had to rush. Anyway, yeah.
Time for just one or two.
I don't have a question, but just a comment. What you see with the major rearrangements in the Hawks cluster, we've seen a similar thing for PAC6. We've moved all the upstream enhancers, downstream, upstream, nothing happens. Absolutely nothing. So there is a common theme which we don't understand.
Right. Yeah, I mean, we can, in the different flies itself, the cluster is split differently. There are some organisms like tunicates in which large genes are spread all over the genome. So in somehow in 3D context, when the time comes to regulate those genes, they come together and they still can talk and make proper body axis. But question still remains then, what is keeping them clustered and they are not moving away? So that's maybe it's something subtle we have to study more to figure out.
Hello, at the back. So in the inversion case where you don't see any change in phenotype, can
we see subtle phenotype, it's not we don't see change, but yeah.
From the image it was very difficult to make out.
Yeah.
Is it the case that these flies, like compared to wild type, will be more susceptible to, let's say, some stress in the environment?
You are very right. I mean, those are the kinds of things. What we are seeing, as I told you, we are only just seeing the cuticle from outside, which we are more used to. Internally, the organs may be, their fitness may be compromised. We don't see whether they are able to fly well and those, none of those. We just seeing some very obvious phenotype that is expected. But you are right, there may be many things because we also know homeotic genes not only do the body axis formation, they have other functions also, which we call as non-homeotic functions. And particularly in vertebrates, there's lots of functions of these genes known. Because their mutation, the way you do genetics, the first phenotype is what you see, the rest of the thing you don't see because they die later on. So we don't understand non-homeotic function, and some of those things may be affected, which we are not scoring in our assays.
Thank you.
Okay, I know. He's going to take it after.
Okay.
So let's thank Rakesh again.